Well, hello, my fellow pre-meds. In this video, we're going to talk about titration curves of amino acids, a high-yield topic on the MCAT, by the way. So, as you're probably familiar with at this point, this is the backbone of an amino acid. So you have your amino group, carboxylic acid group, R group, and hydrogen atom centered around that alpha carbon. Now, notably, for the majority of amino acids, they have two ionizable side chains, and those are the amino group and the carboxylic acid group. And that's why we call them amphoteric. Amphoteric means that they can carry a positive and negative charge at the same time. All right, and then you have some exceptions that can carry three charges, so they can also carry a charge on their side chain. Those are the basic and acidic amino acids, but we're first going to focus on the ones that only have the capacity to carry two charges. Okay, so the carboxylic acid group right there is going to have a pKa, and we're going to call it pKa1, and the amino group will have a pKa2. So this is a titration curve right here. This is the titration curve of acetic acid, a weak acid. We're going to first highlight certain points on this, and then we're going to move on and draw out the titration curve of an amino acid. So as you can see right here, this is the structure of acetic acid when it is fully protonated. So the way titration curves work is that you can start at a very low pH value. So let's say we're starting off at like 0.5 or even less. And then you gradually increase the pH by adding hydroxide equivalents over here. So what you're doing to this weak acid is, as you add more and more of the hydroxide ions, you are pulling away some of those hydrogen atoms or protons from the acetic acid. And then as you increase the pH, you're increasing the amount of acetic acid in solution that's been converted from your protonated form to the deprotonated form, to the conjugate base. All right, so notice here how once you reach this point, which is the pKa of acetic acid, which is around 4.76, once you reach this point, what you have is that in your solution, half of your concentration of acetic acid in the protonated form has been converted to the acetic acid in the deprotonated form, or in the acetate ion. And then you'll notice that here, at this point of the curve, it starts to flatten, it starts to plateau. So despite adding significant amounts of hydroxide ions over here, you see a very slow increase in the curve. And that's because we're around the pKa value of a weak acid, and we call this a buffer zone. So the solution starts to become resistant to pH changes despite adding more and more base. All right, so then you continue to add base over here. You eventually reach a point where all of your acetic acid has been deprotonated, and now all of your solution is in the deprotonated form. All right, so now let's get to the titration curve of alanine. But really, you can swap any other amino acid over here other than the basic and acidic ones. So again, our titration curve, we're starting off at a very low pH, let's say like less than 0.5. And then before we get started with drawing this out, let me just point these out over here. So you have form 1, where you have the protonated carboxylic acid in the backbone, and the protonated amino group, which is carrying a positive charge when it's protonated. And then you have your Zwitter ion, which is form 2. At this point, you have donated the hydrogen atom from the carboxyl group, but the amino group is still protonated. And then finally, form 3, where essentially the amino acid has donated both the hydrogens it's capable of donating, which is this first, the carboxylic acid group right here, and the amino group. Okay, so at very low pHs, you exclusively have amino acids in the form of that first the first form, so form 1. And that's because you're at a very low pH, so both the carboxylic acid and the amino group, relative to the surrounding environment, are acting as bases. So the environment, because it's very low pH, it's donating hydrogens to both the carboxylic acid group and the amino group. So they're both protonated. Now we start to add some more base over here. So we start to deprotonate the carboxylic acid. All right, so here you reach a point which is pKa1. At pKa1, as we saw with acetic acid, you will have equal concentrations of the protonated and deprotonated form. But notice how over here, we're specifically focusing on the carboxylic acid group. So equal concentrations of the protonated form of the carboxylic acid in the amino acid and the deprotonated form. We're not worried about the amino group yet. Okay, so equal concentrations, we also call this point where you reach the pKa1 the midpoint, or the half equivalence point. Now we continue to add base. Again, notice that flattening of the curve over here, and that's because we're at the buffer zone. So as you continue to add base, you reach a point which we call the equivalence point, where now you exclusively have form 2. Because now at this point, the pH is high enough to where all of your carboxylic acid has been deprotonated. However, notably, nothing happened to the amino group. Why not? Because recall, the amino group has a higher pKa. It'll be closer to like 8 or 9. It's more basic. And we're still at a pH of around 6. So none of that amino group has been deprotonated yet, or at least very, very tiny amounts. Okay, so once you reach the equivalence point for these neutral amino acids, that is the amino acids that are incapable of carrying a positive or negative charge on their side chain, you will have reached the PI, the isoelectric point. So that's just something to know. If they give you a curve and they tell you this is the equivalence point, what is the PI? Well, that is the PI. Whatever the pH was at that equivalence point, that is your PI, okay? Next, we continue to add more of that base. So as you can see, we're approaching another flattening of the curve. Well, what does that mean? Exactly, that means we're approaching another pKa value, and that's the pKa of your amino group right here. So pKa2 corresponds to this amino group. All right, so at this point, form 2 and form 3 are in equal concentrations, and that's because notice how the difference between form 2 and form 3 is that you deprotonate the amino group, and when you're at the pKa, we mentioned that you have equal concentrations of the protonated form of the molecule and the deprotonated. But recall that now we're specifically focusing on the amino group. So don't worry about the carboxylic acid group at this point. Okay, and then this would be the second midpoint, or midpoint 2. And then as you continue to add base, you're eventually gonna, so even though the amino group is basic, as you continue to add base to the point where now the pH of the solution reaches like 10 or even 12, at this point, the amino group will be relatively acidic compared to the environment, and every single amino group in the solution will be deprotonated, and you will have exclusively the concentration will exclusively be form 3 of the amino acid. So now let's move on to the titration curve of acidic amino acids. So recall these include glutamic acid and aspartic acid. Now we won't draw it out in as much detail. I just want you to notice common themes with this curve and compare it to the basic amino acids, as we'll see in a bit, because sometimes they'll show you a curve and they'll tell you, is this a basic or acidic amino acid just based on the curve. So first of all, you can sort of look at the 4 forms we have over here. So you have the first form is going to have a protonated amino group. Notably, that R group doesn't have a charge yet, right? Even though we said that it's capable of carrying a charge, and that's because in the case of the acidic amino acids, the side chain has a carboxylic acid group. So it'll only gain that negative charge once it donates the hydrogen atom. So at low pHs, that carboxylic acid will still be protonated and not carry that negative charge. So I just want you to notice over here that you have pKa1, pKa of the R group, and finally pKa2. The PI of these acidic amino acids will be sandwiched in between the pKa1 and pKaR. This is very important. When you are trying to calculate the PI of an acidic amino acid or figure it out based on titration curve, you're going to go based on pKa1 and the pKa of the R group, and don't worry about that pKa2. Now let's move on to the basic amino acids. So once again, you can pause the video and take a look at the difference over here with what you see in the different forms. So we have 4 forms. Notably, that side chain is positively charged because these contain a positively charged, these contain an amino group in their side chain, so they're capable of carrying a positive charge on that side chain. So notice how over here you have the PI sandwiched between pKa2 and pKaR. So to get the PI, you either add those and divide them by 2, or you just use this titration curve if they provide you with it, and you take the point that's in between pKa2 and pKaR. Now notice a huge difference where you can just glance at the curve and tell if this is acidic or basic. You'll see that you see this dramatic increase right here? It happens pretty early on, whereas over here, that dramatic increase in the acidic amino acids happens later on. So the PI happens earlier, and then you have the dramatic increase, whereas in the basic amino acid titration curve, you have the dramatic increase, and then the PI happens later. So this is a way you can just differentiate between the two amino acids just by looking at their curves. This is definitely one of the more tricky slide decks, so if you have to watch it again, download the slides and annotate as you're watching it. And if you have any questions, you know where to reach me. If you found this video helpful and like the way I teach, I'd recommend watching the rest of the playlist. I spent a lot of time making sure that the videos build on one another, so it makes sense to watch them in order. Also, don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow, and I would really appreciate it. Also, did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide decks. Finally, these videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing Pre-Med Companion Discord community. Thanks for watching, and I'll see you in the next video.